Hi, everyone. Today we have as our guest Erik De Schutter, who trained as a neurologist, but fortunately for us, decided on a career in scientific research as a computational neuroscientist. In the 1990s, while at Caltech with Jim Bauer, he developed the first detailed Purkinje cell model. He then started a laboratory at the University of Antwerp, Belgium, and in 2007, he moved to the Okinawa Institute of Science and Technology, where he has been since then. In 2013, he co-authored a highly influential paper proposing that synchronous spiking can be used to multiplex temporal and rate coding. Today, he's going to concentrate on the dendritic computations in the Purkinje cells, suggesting that each branch may be an individual unit of computation.
Okay, thank you for the invitation. I'm happy to be here. So if you just look at this first slide, you maybe understand why I moved to OIST, right? But that's a joke. It's actually a really fantastic research environment. Also multidisciplinary, which for someone like me is really home, right? To be in a multidisciplinary place. But I'm not going to be talking about OIST today. We're going to be talking about our recent modeling work in the Purkinje cell. And so this is kind of the overview of today's talk. So I'll introduce a bit. I'll talk about why we're interested in pulses, introduce the model, then talk about parallel fiber-evoked dendritic spikes and what they look like in the model. And then the big part is about a new updated version of the model we made that is actually just published in iScience last week. And I'll describe that in detail, and then also talk about some unpublished work where we've also looked at feedforward inhibition in this context. So first of all, a big focus of today is going to be pulses in Purkinje cell simple spiking, and why are I so interested in these pulses. And this is this paper as I mentioned. So this is basically us analyzing data from Peter Teer's lab, so monkey recordings of the saccadic eye region of the cerebellum, using techniques which we developed previously. And this is really looking in detail at simple spike firing of single neurons. And in 2006, we published this study where we basically observed that in rodents then, that you can think about the Purkinje cell spikes as two kinds. So it's a spontaneously firing neuron, right? And when it kind of fires spontaneously, it fires fairly regularly. So the interspike intervals don't vary a lot. And of course, the firing rate can change, and that's controlled also by synaptic input. But sometimes this kind of fairly regular fast spiking is interrupted by what we call a pulse, right? And so you can then identify the pulse initiating and pulse terminating spike. And if you now look at that in Purkinje cells in the saccadic eye region, and you correlate it with the start of the saccade, so this is eye velocity, and so the saccade starts here. First of all, this is also linked to very clear activity in the local field potentials, suggesting that there is a lot of synchronized activity ongoing. But in the Purkinje cells they recorded from, you see that there is really a big increase in pulse initiating spikes just at the onset of the eye movement, right? And so in other words, we think that this pulse basically, which means that you kind of stop inhibiting the cerebellum nuclei, that this is a trigger for the motion, for the initiation of the movement. And more recently, Indira Raman, for example, in the whisker system, has been talking about what she calls synchronous spike suppression, but which I really think is just a different way of analyzing the data, but it's basically a similar finding, in my opinion, kind of synchronized pulses driving involved in the whisking. Abigail Person also has a preprint online in the reaching movement, where she again reports similar findings in the context of reaching. By the way, the regular spikes also do something they basically are correlated with eye velocity. But that's not really the topic of today's talk, but it tells you why we're interested in pausing. And then ask ourselves, how do you actually generate such a pause in a Purkinje cell, right? Obviously, you could do it with basket cell inhibition, but is that the only way, or are there other ways of generating pauses? And that will be largely today's topic. So we approach this with computer modeling. And so, Reza mentioned this model I developed in the early 90s. But we realized that gradually this became outdated, right? There was lots of new data, especially about the ion channels available. And so it became time to generate a new model. And Yungyang Zhang, who was a postdoc in the lab, is now professor at Tsinghua University in China, spent basically 2 years of his life developing this new model, hand-tuning it. And then we got this nice publication, first publication about it in Cell Reports. And I'm not going to explain everything here, but I only want to point out one thing here. So this is a picture of the voltage during a complex spike. And you notice that, and so the difference between these three is the holding potential, at which membrane potential you hold the cell. And you see that that is strongly depends on holding potential. Also, the duration of the complex spike depends on this holding potential. But especially that there is, for these lower holding potentials, where you do not get a full-blown complex spike, that there's a lot of heterogeneity in this Purkinje cell dendritic tree. And that's basically going to be the theme of today, right? So the morphology of this cell really changed a lot how it responds to synaptic input. And you already see that in the complex spike. So this model is kind of a standard compartmental modeling for the people who are kind of more interested in the technical side. So we use a neuron simulator to simulate that. And as is often done in such models, so the big work, what Yungyang spent almost 2 years on, is figuring out channel densities, because at present that's still not really quantitatively measured in experiments. And so you have to fit those, and to make that work simpler, you do not normally do this in every individual branch, but you say, okay, I assume it's the same for all spiny dendrites. Right? And so that's why we call it a homogeneous Purkinje cell model now, because basically it has these homogeneous channel densities across the dendrite. Now, making such a model is great, and in this case, we show that it actually also nicely replicates somatic firing. It replicates the complex spike, but it's being fitted to do this. And so it's nice if you can show that this model actually has predictive power. And so there were 2 follow-up papers where we basically showed that. And the first one is this. I'm only going to show this slide. There's much more in the paper, but this is about the phase responses of the phase of the Purkinje cell. And the phase response is basically where you have it firing regularly. You inject a bit of current and you see how the timing of the next spike changes. And you then do that at different phases during the interspike interval. And you kind of plot that in a plot like this. And so what was known experimentally, actually, Mike Hauser showed this, is that this is strongly firing rate dependent in Purkinje cells, right? So the firing rates are shown here. And so at low firing rates, there's hardly any modulation, and then it goes wildly up. And you can plot that as this amplitude versus rate. And that then looks a lot like what you observe in experiments, right? So this is experimental data of the same paradigm. And so basically the model was not fitted to reproduce this. And actually previous Purkinje cell models, including 194 model, could not reproduce this phenomenon, right? So this is really nice, right? Where you kind of test it on something new and it works. And then the next one is then about dendritic calcium spikes. So again, work by Mike Hauser and Ede Ranks. And actually we use in the model the morphology reconstructed by Ede. And so they showed that when you synaptically stimulate parallel fibers on a particular part of the dendrite, so in this case, this is the thread trace, that you have this kind of threshold phenomenon, right? So for small stimuli, you get an EPSP, and if you go above a certain threshold, you get this very sharp spike. And in neighboring dendrites, you don't really observe this so much. There's a big amplitude difference. And then if you do the same in calcium imaging, where here they stimulate synaptically and here electrically, you see that with the synaptic stimulation, you get, again, this very big calcium spike, which does not actually affect much this red region, right? And vice versa, with current injection, you just get something that looks more like an EPSP. So this shows that you can generate, with clustered parallel fiber input, localized dendritic calcium spikes in the Purkinje cell. So we wondered whether you also observe this in the model, and yes, you do. So this was the final paper you published with me. And so basically we kind of subdivided, and we've kept this subdivision also for the new study, the neuron in what we call branches. And so the idea is the black is a smooth dendrite. Whenever you've got the root of a spiny dendrite branch, so that is called a separate branch. And basically we identified 22 branches in this mouse morphology. And so then when you put, so the green dots here are parallel fiber synapses on one of these branches, branch 8, and stimulate, you see that as you increase, so with low number of parallel fibers, you just get EPSPs, which become bigger and bigger. Now at some threshold, you now generate a dendritic calcium spike. So this is very similar to what Ede showed. So these data are plotted here. And so if you just plot that in a plot like this, where you take the peak amplitude versus the number of synapses, we see that there's a region where you get a linear increase. So that kind of corresponds to what Kamera and Kodak showed many years ago in slice experiments, that you have linear responses to synaptic stimulation. But then at some point, you cross this threshold. At the threshold itself, it's can be either, and above this threshold, you have now kind of a fairly constant amplitude of a big dendritic calcium spike. So this is kind of, again, the same thing. Here we actually show that you can block this with hyperpolarization, so not strong hyperpolarization, just hyperpolarize the cell a bit somatically. And you see that now at this parallel fiber strength, you no longer induce a dendritic calcium spike. But why I really show this is to show you the underlying ionic currents. So these are plotted here. So the big one that triggers this is a P-type calcium channel. There's also T-type in the model, but it's not really involved in this phenomenon. So it's completely P-type calcium driven. And then there are a bunch of potassium channels which repolarize the dendritic spike. But among them, KV4 is basically the one that activates fastest. That first activates, and that will be important in the next study I'm going to be talking about. And then finally, so the title of the paper was multiplex coding, so referring to this study I started the talk with. And so indeed, that's now what you kind of see when you look at somatic recordings. So basically, this is the spiking in the soma, and we provide some random background synaptic input to basically make the spiking at random times in the absence of synchronous input. And then when you do the synchronous branch stimulation, you see that this basically in the peristimulus histogram starts to generate a peak, so which corresponds to a burst of spiking. And this peak increases as you increase the number of parallel fibers, so that's a linear increase. At the threshold, you see it can either dendritic spike or not. And you start to see what then in there I would call spike suppression after the burst. And then above threshold, you really clearly see you have a burst pause sequence, right, with a very pronounced pause. And that's also very visible in the peristimulus histogram. So basically, we could generate this in 4 branches of this Purkinje cell model. So that actually brings me to kind of the problem with this model. And then, let me first summarize this, and then I'll come back to that point. So this is kind of the graphical abstract of that paper. And so basically, we see that you can have either just linear coding, where you have a constant increase in the amplitude of the EPSP, which just generates bursts in the somatic output and in the peristimulus histogram. Or you can have, so in this model, this is the case for most of the branches. And then in 7 branches, you can actually have this transition where you have what we call a bimodal response, so either initially linear increase and then this jump to this bigger amplitude with the dendritic calcium spike. And when you are above that, you get this burst pause sequence, right? And so basically, in other words, the what you kind of see here is this multiplex coding, right? You can have just purely changes in firing rates, or you can have also this pause. Right? But as I just mentioned, so one issue with this model was that we actually saw this bimodal response only in some of the dendrites. And so this is now the topic of the new paper which just came out last week, which was the work of Gabriel Sertalla, a very gifted postdoc in the lab. And this kind of then summarizes again what we just saw from the previous paper. So but now, so in the previous paper, we really focused on the ones where you got the bimodal response. So for example, in this branch 5, but in branch 18, which is over here, you just only get the linear response. You cannot, with the standard homogeneous model, you cannot evoke a dendritic spike at, we went even higher than what's shown here, right? And then also, as a control, we now plot actually a branch which is not stimulated at all, so branch 12, and you basically see that nothing happens there, right? So it doesn't receive any parallel fiber input. So basically what Gabriela set out to do is to kind of, so we believe that this is probably not physiological, right? That only some branches can generate dendritic calcium spikes, and it is probably an obvious consequence of the fact that we've put the channel densities identical in all these branches. So what she set out to do was to kind of start changing channel densities in branch-specific ways and see whether we could then solve that problem, basically. And the obvious candidate to look at is here the P-type calcium channel, right, which really drives the dendritic spike. So this is branch 3, and which initially just has only a linear response. And then Gabriela increased the density of the P-type channel by 30, 50, or 70%. But with 30%, you already see some effect, but with 50 and 70% increases, you really see a very solid effect and a nice bimodal response. And you see that this also affects basically the threshold, right, at which you generate the dendritic spike. This is at this distal point. In this branch, actually, there's not much of a response in the proximal, close to the smooth dendrite. That is not true for every branch, as we'll see. So this is also an example that's highly branch specific. So this is kind of the summary data. These are then what actually the dendritic spikes look like. So initially EPSPs, and at 48 parallel fibers, we have the threshold for dendritic spiking. In the proximal point, you don't see much in the distal point. We can look at a movie of this thing happening, and you see that it's nicely localized. So it's localized here to this region. And then it generates a somatic spike. And then And then as expected, so with the dendritic spike, so if you're above threshold, you actually get a burst pulse sequence, right? And below threshold, you just only get a burst. Again, quite comparable to what we've seen before in Yang-Yang's paper, with the difference that this branch in Yang-Yang's paper did not generate dendritic spikes. We fixed that by increasing this number of P-type calcium channels, the density basically in this branch. So that's one change we made. I'll show you a summary data of all the branches a few slides further. I first want to discuss about another change we made. So in some of these branches, so this actually the calcium spike actually does not stay localized, right? It spreads around in neighboring branches. And it's actually mainly a problem at this left main division of the dendrite. That's really very pronounced. And Gabriella looked at how she could fix this. And the logical candidate about there is to think about this KV4, which is basically an A-type potassium channel, because that's the one that activates fastest. And so let me show you the results of increasing now that KV4 potassium channel. And I'll show you that with movies. So this first shows the original, so without changes. So that was we saw static images of this spread in the previous slide. And then now we increase the KV4 in that branch and also in neighboring branches. And it stays now really nicely localized. Right? So basically we could fix the spread. Unfortunately, we could not do this completely everywhere. So this big branch, it was originally spreading into branch 8, shows a similar problem. And there we only could partially fix it. So this is the original case where you see it spreads so branches 7 and 6 and even 5. And then with this increase, you'll see that there's still a bit of depolarization here, but it doesn't go into the red anymore. It stays kind of in the light blue range. Right? So it's a much smaller depolarization than before, but there's still a bit of spread. So again, showing, and this will be kind of the theme of today, right? The morphology really matters, and all these individual branches react differently. With changing channel densities, we can make them look much more similar to each other than in the homogeneous model, but there's some things you cannot fix completely, right? Morphology is stubborn, and with whatever channel density changes you make, in this case, you cannot fix this spread to these neighboring dendrites. At least you can reduce it, but not completely eliminate it. So this is then the summary. So this was originally the original Yang-Yang's model, where you see that just 7 branches showed a bimodal response. The grays are all the ones that only shows linear increases. And then this is Gabriella's heterogeneous model, where you now see that we have solid bimodal responses in all 22 branches, but not the differences in parallel fiber thresholds. Right? So there's still a huge spread in parallel fiber thresholds. And I showed you earlier in the first example that we can shift this a bit, but we're limited to how much we can shift this, because if you increase the P-type too much, then the spike will start to spread again, right? So there's only limited range in which you basically can shift this around. And so that's summarized in these bar graphs here. So these are the changes in P-type calcium channel for all 22 dendrites. And notice that we changed in most of all 22 branches. And notice that we changed in most of the branches. Right? For example, in branch 8, we reduced it. Right? So that was the one that still kept spreading. And it was also one of the ones that was originally already generating dendritic calcium spikes in Yang-Yang's model. And in all the ones that were not causing spikes, there we had to increase it. Then we increased the KV4 in this region we were discussing before, and also a few of the branches in the middle region, but much less. And this then summarizes the parallel fiber thresholds, which we already saw in the previous slide, with kind of the mean in this broken line. And you see that, yeah, it's basically extremely variable. Right? So this variability is caused by morphology. And so we also wondered whether basically specific morphological properties could predict this. And the only relationship we could really establish is with the P-type calcium channel densities necessary to generate the dendritic calcium spike. So this, relative to the original value, which is this blue, not really light and not really dark blue, you see that in most places we have to increase it, and that goes to the yellows and the browns. In some parts we actually decreased it, and that's this dark blue. And then we could correlate this basically with two morphological properties. One is the total length of the branch, which is basically the summation of the length of all individual sub-branches, right? And you see this varies a lot, right? So the very small ones and the very large ones, like 21 and so on. And basically the large ones require decreases in P-type calcium densities, while the shorter ones require large increases. So it's inversely correlated, and this was highly significant. What was also significant, though less, is the distance between the root of the branch and the soma. So again, the ones that are further away from the soma tended to have lower P-type calcium channel, while the ones that are much closer to the soma tended to have bigger ones. Though notice this is not completely strict like, because these three, the highest ones, are kind of midway, right? So that was so there is a statistical relation between morphological properties and at least this, but it's there's a lot of scatter still. Right? And then finally, we also wondered what would happen when we co-activate branches, right? So we activate two branches together. And this again turns out to be highly specific on which branches you co-activate. And so that's summarized. I first show an example where it's fairly simple, where nothing special happens. So branch 5 and 10, which are well separated, we'll see that soon. And so branch, if you only activate one of them, this is this arrow. So the threshold for single activation of branch 1 is here. Branch 5, sorry, is here. Branch 10 is here. And then with co-activation, it's a bit shifted to the right, while conversely for branch 5, it's a bit shifted to the left, but it's still quite similar to the original thresholds. And basically, you only activate these two branches, and you see that they're nicely in these color plots. So you get this nicely localized activation, and with a bit of effect on their thresholds, right? But not much interaction in essence. This is a completely different example. So these are neighboring branches, 11 and 12. And you see, first of all, their individual thresholds are much higher than the combined threshold. And you also see actually 5 different branches activate here, right? So you stimulate 2 and 5 activate. So this really spreads like crazy, right? And that's what you see here. So these are the two we activate. It already first spreads to 13, and then also to 9 and 10. Right? And this is in the middle region of the morphology, and it's very basically pronounced here. Right? So this multi-branch activation, basically what from these two examples you see that you can get a lot of different effects. And so we kind of, Gabriella did this very systematically for all possible combinations. So branch A combined with branch B. And we looked also at how this relates to the main divisions of the dendritic branch. So we have this left one, which is then in cyan, the middle region, which is in orange, and the right, which is in gray. And we colored the ones belonging to these in these rectangles here. And you see that that really gives kind of structure to this data, right? So here we're looking at the thresholds, and the red triangles basically means that the combined threshold is lower than the single branch threshold. So that was the second example we saw. And we see that you indeed get that mainly when you activate branches within the same main division, and also kind of much more in the gray and orange division than in the light blue one. Right? So it really depends on where you activate. Basically, if you activate branches which are neighbors on the same subdivision, then you see this change in threshold a lot. But it's quite variable. You see some cases also out here, right? So it's not strictly, it's not a strict rule. Then the blue is where the threshold doesn't change, and the black where the threshold actually becomes larger, right? And that's mainly then for just combining branches on different subdivisions. And then the propagation we saw. So this propagation is mainly occurring in the orange and gray subsections when you co-activate branches in the same subsection. And so this kind of codes for how many branches get co-activated from 1 to 3, and the rate is more than 3. So you see that really on this gray one, you get a lot of big co-activation. In the example we saw here were exactly 3, because basically there are only 5 branches here. So it cannot be more than 3, and so on, right? And so black is no spreading at all. And again, you see that also outside that there's some cases where this happens. So it's there you see some clear tendencies, right? So within these same divisions, but there is a lot of stuff also happening that you cannot capture with simple rules, basically, which has to do with really the individuality of these branches and their individual properties. So this is then everything I wanted to tell you about our recently published paper. In the last few slides, let me talk about some follow-up stuff Gabriella has been doing with Taiwanese intern, Pinju Chu. And so now more focusing really on the pulses. Right? And so this just kind of summarizes what we saw before, right? So this is the dendritic. So this is a case which originally did not generate dendritic spikes, but with increasing the p-type calcium channel, it does. And so in the original homogeneous model, you only got these linear responses, no pulses. In the new heterogeneous model with increased p-type calcium channel densities, you now have this burst pulse sequence. And so we want to look at this in more detail. And we worked very hard. Gabriella generated lots and lots of these peristimulus histograms, which are basically based on 500 runs of the model. And discovered that they're really difficult to do statistics on. Because how do you really define a pulse and the pulse duration? And like if you have 500 runs, maybe in one of those 500 runs, actually you get a spike smack in the middle of what you think is a pulse. And then do you still call that a pulse or not? That's an easy one. If it's only 1 out of 500 tries, you say, let's ignore that, it's a pulse. But there are a lot of these border cases where, you know, it just is very difficult to have a consistent rule where you can say, okay, this is the pulse and the rest is not a pulse. So we basically went back to the approach we did in the eLife paper, which I started the talk with, which is analyzing ICIs. Right? And so here we're looking at the duration of the longest ICI after the short window after the burst. And again, in black, there's basically no burst. So you see a small increase in the ICI duration, but for the one that generates the burst pulse sequences, you see that above this parallel fiber threshold, you have a really strong effect on the duration of the interspike interval, right? Which becomes significantly longer. and we then kind of fit a curve to this. So in the rest of this presentation, we basically are going to look at this because we can kind of statistically analyze it, and it's very consistent, right? And it's basically a single trial approach to the things, right? Okay, so if you now look, so this is a heterogeneous model we presented in the paper, and now we ask the question, do we get pauses on all 22 branches? So on most of them we do, and these are examples of them with the PSTHs. And again, for the ICI analysis, but basically now the yellow zone is below threshold, the gray zone is above threshold for parallel spiking, for parallel fiber evoked dendritic spikes. And so you see a subtle but significant increase here, and in this case, a really big increase in ICI duration in the gray zone, right? So the dendritic spike really prolongs the ICI. But then in some of them, you basically observe this, right? So no bursts, but no pauses, and no real strong ICI modulation if you plot it. So there was in 3 branches in total, but I'm just going to show you 2. And so again, we went back to the previous approach to try to fix this by changing channel densities. And basically, based on a lot of evidence, we looked at the SK2 calcium activated potassium channel. So this is well known to be actually modulated in the, so Christian Hansel has published several papers where they showed that this is the basis of what they call excitability plasticity. Right? So this is being modulated by learning in Purkinje cells. And basically, these were these 2 examples I showed in the previous slide, which originally did not show pauses. And now if you, so that's the black traces, and if you double or triple the SK2 channel density, you now really get a big increase in ICI durations in the presence of a dendritic calcium spike. Notice it doesn't change much in the linear range, right? In the linear range, they largely overlap, but when you have this dendritic calcium spike, you now get this much bigger, much longer ICIs corresponding to a pulse. If you look at the dendritic calcium spikes yourself, actually the effect is quite modest, right? So there is a bit of a more pronounced after hyperpolarization with the bigger SK2 than with the standard value, but it's a modest effect. So, but it has such a strong effect, we believe, on the interspike intervals because it's a long during effect, right? So it really lasts quite long, and that generally is quite effective. Then we also wondered what feed-forward inhibition would do to this, and this is what Purkinje has really been working on. And so feed-forward inhibition here is modeled very simplistically. So we assume that it arrives on the same branch as the synchronous parallel fiber input with a 1.4 millisecond delay, right? And then we modulate the number of synapses, inhibitory synapses we activate. So that's a free parameter, how many inhibitory synapses are being activated. For the rest, it's just linked to the synchronous parallel fiber input. And actually, Jung Liang already did that in his paper. I haven't shown that, and showed there that inhibition shifts the threshold for generating the dendritic calcium spike. And so we will now, in this heterogeneous model, look at these 22 branches. We observe that clearly in some, like for example, branch 21 is a nice example, right? Where you see that as you go from no inhibition to half inhibition to full inhibition, you basically shift this curve to the right. There are other places where this happens. 22, it's clear. 18, a bit less pronounced. 12, and so on. But in a lot of branches, you actually see hardly any shift at all, right? So this is again an example where actually this effect is extremely branch specific, right? So it's just and that's something we didn't observe really in a homogeneous model. Though we only looked there at the branches that could generate dendritic calcium spikes. And that then also, I mean, this shifting of the threshold then also raises the question, okay, how is this now going to affect these pulses, right? So that is then the last slide I'm going to show. So that's basically the effect of this feed-forward inhibition on pulse duration. And so if you look at this is in the linear range. It always increases the ICI duration, which we take to be corresponding to the pulse in a range of about, let's say, 2 up to a bit over 6 milliseconds, right? It increases ICI duration. So inhibition is always effective. It's also always effective in the dendritic spark region. But if you now compare the two, right? So before we said, okay, you did not have a pulse with in the linear range, but a pulse in the dendritic sparking range. So if you now compare these two ICIs, in the presence of inhibition, you actually get a very variable picture. And that's what we've done here. So blue are branches where combining the two makes the pulse in the dendritic spark region even longer than before, right? Compared to before. Compared to non-dendritic spark. Red are ones where the opposite happens. Actually, the dendritic spark now makes the spark shorter. It's generally speaking not such a big effect, but in two branches it's really pronounced, right? So that's kind of really interesting, right? So if you combine the feed-forward inhibition sometimes enhances the pulses generated by the dendritic calcium spikes, but in many branches it actually masks the effect. And so that's kind of a quite complicated interaction. And again, it's we color coded here on the branches. And so you see, again, it's a really branch-specific property, and it's not easy really to predict where in the morphology it's going to happen, right? Some of them are really close to the soma, some are more distal. You find cases in each of the main subdivisions, so it's really difficult to predict, right? And that is basically brings me to my conclusions. So morphology basically always matters. Even if you kind of optimize channel densities to make the branches look as much similar to each other as possible, you cannot erase all the differences, right? The branches keep their individuality. So to kind of summarize what we saw, so in the original homogeneous channel density, only a few branches could generate calcium spikes. By increasing the p-type calcium channel, we could enable calcium spikes in all branches, but with very variable parallel fiber thresholds. So this one form of variability between the branches. These spikes are usually localized, but that depends on the KV4 channels. And basically, if we had to increase them in some places to keep localized. I didn't show you that if you do the opposite, if you decrease KV4 channels, things spread like crazy. Multi-branch activation is extremely dependent on which combination of branches you activate, and mainly on whether they're on the same main division of the dendritic tree or not. Then in some branches, dendritic calcium spikes did not generate somatic pulses in the original heterogeneous model, but if we increased SK2 calcium activated potassium channels, we actually could generate pulses in all the branches. Conversely, if you decrease it, pulses will start to disappear. And then finally, feed-forward inhibition always increases the duration of the ICI generated by the parallel fiber input, but it interacts in an unpredictable way with the dendritic calcium spikes, in that it sometimes enhances the effect and sometimes it masks the effect, right? And so again, all of this is extremely branch specific for this mouse morphology. And that's basically everything I wanted to tell you today. So I'm open for questions.
Thank you so much, Erik. And yes, we're open questions. So please go ahead and just raise your hand and we can come to a question. So I'll start out the question. So Erik, you showed that as input strength in terms of number of parallel fiber synapses increases, there's this nonlinearity in the dendritic response, which produces increases in spike rates, but also then a pause that follows that. Can we interpret any of this in terms of the same parallel fibers going to nearby molecular layer in our neurons that inhibit that Purkinje cell? And could this be interpreted in terms of this network effect, not just effect on the Purkinje cell itself, but a network effect on the Purkinje cells?
Well, that's what we try to explore with looking at this feedforward inhibition, right?
Yeah.
Now, of course, again, I should emphasize that our model of feedforward inhibition is not very sophisticated, right? Because we're really only inhibiting the branch which we're also activating, while in reality, if you think about how the axons of stellate cells go, they probably would also inhibit other parts of this dendritic tree, right? So there is space there to become more sophisticated about how we do this. But in first, at least with this simple approach, we already see this huge diversity, right? And this kind of non-intuitive interaction that in some cases actually inhibition masks the effects of the dendritic spikes generated by the parallel. Right? It's so this is going to be quite complex in reality, I think. Yeah, of course, I should also add, so the one thing which we're not doing here at all, so we've been changing densities of voltage-gated channels, we're not changing the synaptic strengths, right? So we're increasing or decreasing the number of synapses we activate, but we're not changing the strength of these synapses. There's, of course, another dimension you could do things with, right? Which the cell probably does.
Yeah. Okay, Anders, go ahead, please.
Yeah, hi, thank you, Erik. You can hear me, right?
Yes.
Yes.
Good. Yeah, so I mean, maybe sort of backing up a little kind of question, but when you talked a lot about what happens in different branches and so on, and one thing I often wonder about is how much does it matter? I mean, obviously you have these Purkinje cells with massive dendritic trees, but then in the end it boils down to the spiking rate in the Purkinje cell. So what's your reflections on that?
Well, so basically I have two. First of all, let me end with the last part, right? I mean, so what I kind of showed in the first slide is that, in my opinion, it's not just firing rate, right? You have these, when a bunch of Purkinje cells pulse synchronously, this will have a big effect on the cerebellum nuclei, downstream cerebellum nuclei neurons. And so this is, of course, I mean, if you do a firing rate measure, you will see what in Dirac calls spike suppression, but that's kind of not a very accurate way, let's say, of looking at it, right? So there's more than the firing rate. That's what I'm trying to say. But then whether these branches really matters, that comes back to what I mentioned in the introduction. Well, we were actually discussing this before I started the talk, so I didn't mention that actually during the talk. So that's the central question there is whether there's clustered parallel fiber input or not. If you believe Mike Hauser, he published a nice study demonstrating a few instances of clustered parallel fiber input. It was a very challenging experiment, so it's a small sample, but he showed it. Conversely, more recently, Angus Silver kind of imaged parallel fiber activity, and he claims that there is no evidence for clustering at all. Right? And I think that is kind of the real challenge in the field. I mean, it's been the real challenge for my entire career in cerebellar, thinking about the cerebellum, is that we really know very little about what parallel fiber input really looks like. How is it structured both temporally and spatially? Right? Is it clustered? Is it not clustered? Is really this strong synchronization or not? There's really no good data about that. And as long as you don't know that, I cannot answer your question whether branches matter or not, because it depends on how the synaptic input is structured, especially spatially in this case.
Thank you.
Thank you. Erik, Sasha here has a question for you.
Hi. First, I wanted to say that was really fantastic. But one thing I was curious about was, how did you distribute the synapses spatially within the branches? Was that informed by any prior work that had been done experimentally, or was that just arbitrarily?
No. It's basically a random distribution. So they're randomly distributed within the extent of this branch. So there's we don't really have data.
I see. So was that shuffled then on a trial to trial basis?
It was shuffled in another way, which actually turned out to be. I mean, that's how Yang did it originally. We kept doing it, but it has clear disadvantages. So if you increase the number of parallel fiber synapses, it's not that you add additional ones; they all jump around.
Yeah.
They all change location, right?
Yeah. So the reason I ask is because you talked about looking at the PTET calcium channel and KV4 densities as a function of like morphological aspects. But I was curious if you looked at like passive synaptic integration as something that might have affected that, where if you have your synapses randomly placed and they all end up being either relatively close to one another or relatively far from one another, they could be arriving at the root at different times and affecting how those spikes are generated.
It would, I think, I mean, it will depend a lot on how big the branch is, right? For these small branches, I really don't think it matters that much. For the bigger ones, indeed, there could be some effects there. And that is the other criticism you could kind of phrase here, right? I mean, the way we define branches is kind of arbitrary. I mean, well, it is a nice, strict mathematical definition, but from a biological viewpoint, maybe the cell doesn't care about that at all, right? It's
yeah. I was because you guys looked and saw these really nice trends with branch length determining what you were gonna get with those channel densities, and I was wondering if the synapse placement might be a factor that was driving that as well.
Well, it's for sure if you would concentrate them very strongly, for example, on the distal end, then probably you're going to be changing the threshold for evoking the dendritic spike. That's what I would expect. But we didn't explore this really in detail. And well, I can tell you that this is still unpublished, but we actually have data about spine location on a mouse Purkinje cell. And it turns out to be the densities turn out to be quite heterogeneous and localized. So the places where the densities are extremely high, and then close by you can have suddenly much lower densities. So this kind of uniform random placement in that sense is not correct, because actually, if you look at the spines, they're not uniformly placed. They have very irregular densities.
Awesome, thank you.
Any other questions for Erik? Hi, thank you for the very interesting talk. I was wondering if you can describe, so given this information, the stellate cells and basket cells should play very different roles, because one of them are changing the dendrites and one of them is changing the soma. I'm not sure about it, but I think Mike Hauser had a paper showing that the MLIs, the further they are from the Purkinje layer, the delay of the inhibition doesn't change, but the amount of inhibition changes. So given the fact that both of them are probably 1 millisecond inhibition,
how do you think that affects the computations that are being done between
Well, so we actually published many years ago a modeling study with my old 94 Purkinje cell model where we compared these two, and it's true. So I mean, basically, the basket cell is a very strong inhibitor of somatic spiking, right? It just kind of shuts down somatic spiking in a very predictable way, and while the effect of stellate cells, so that was without looking at dendritic spikes, right? So it was just looking, and so the effect of stellate cells is much more subtle and in a way more kind of, let's say, fine red modulation, right? And that so what that is, I think, the summary, right? So you have one type that really inhibits the spike initiation zone, so that's going to have a very strong effect on spiking. Another one that kind of more sits, so that's what we suggested in that modeling paper. It more kind of changes the average dendritic membrane potential. In the Purkinje cell, the dendrite is, except during the peak of the spike, of the simple spike, but it always, in other periods, the dendrite is always more depolarized than the soma. Right? So it actually provides a depolarizing force to the soma. And you can kind of modulate this amount of depolarizing effect with stellate cell inhibition, right? If you increase inhibition, you will reduce this effect or not. So that's one way of looking at it. Here we then kind of look additionally at the interaction with dendritic spiking, which we didn't do before, right? So that's kind of a new angle on it. But yes, just, I mean, because of their location, this is, I mean, there's a long tradition of looking at the effect of the location of synapses in modeling. Actually, Christoph Koch published some seminal papers on that during his postdoc career. And in general, if inhibition is much more effective if it's between the spike generation region and the excitatory input, right?
Yeah.
So the basket cell is the core example of that, actually, right?
Exactly.
Yeah.
So do we think functionally there is a difference? Because like you're showing that these different branches are doing different computations. Are stellate cells targeting to like modify those computations for each individual branch versus basket cells are in general just like controlling the output? Or
yeah, I would say that the second one is very clearly true. The first one, as I mentioned, the simplification we made here is the assumption that inhibition would target the same branch as the parallel fiber excitation. And if you know the shape of stellate cells and their dendritic arborization versus their axonal arborization, that's actually probably not entirely true, right? So the stellate cells will probably also inhibit some branches that we were not synchronously activating with parallel fiber input. So in reality, it's more complicated than what I showed.
I see.
But you know, that's, let's say, future work, and also the difficulty there is actually the data sets of combined stellate cell morphologies with Purkinje cell morphologies is not exactly large, right? So it's kind of not really easy to come up with a well constrained connectivity scheme to model this. No.
Thank you. Thank you so much, Erik. Thank you so much.
Okay, so thank you for listening in, and And now, okay. And I'll be happy to come back a few years hence with another story. Right?
Wonderful. I hope to see you soon. See you,
Yeah.
everybody. Bye. Have a good Tuesday.